Good afternoon. This is going to be a Camtasia recording of a supplement for the third quiz, Topic 3, Cell Structure and Function. We're going to focus on the organelles and their structure and their function. I'm doing this so that we can save a little bit of time in lecture. This material actually is pretty much straight descriptive material. There are a few concepts that we're going to throw in here as we go along, but mostly it's kind of descriptive. So I think actually doing this through a Camtasia recording is a reasonable way to get this information across to you. You're going to want to go through this information to prepare yourself for Quiz 3, and you'll notice that in the wish list of concepts for Quiz 3, describing what the organelles do is one of the requirements that you should be able to fulfill. So here we go. Pansy Answer Dog is here, and she is alert and ready to answer any questions I might have. So there are two types of organelles. We're actually going to cover this in class as well, just this first couple of slides, but I think it bears repeating. There are those that have a membrane. Those are membranous organelles. And then there are those that don't have a membrane. Those are non-membranous organelles. And I think it's reasonable to sort of separate them, and we'll see some interesting things about the membranous organelles. Excuse me, here is a list of the membranous organelles: the nucleus, endoplasmic reticulum. There are two kinds of endoplasmic reticulum. There's rough endoplasmic reticulum, and there's smooth endoplasmic reticulum. We'll talk about each of these. The Golgi apparatus, or some books will refer to it as the Golgi complex. We also have lysosomes, peroxisomes, and vacuoles. Mitochondria are also a membranous organelle. We're going to see that some of these are associated with each other and others are independent. The mitochondria are independent. For the non-membranous, we have a nucleolus, the ribosomes, centrosomes, which actually contain centrioles, the cilia. Cilia is plural, usually they occur in groups, and the flagellum, which usually is singular. Flagella would be plural, but typically you don't see multiple flagella. The cytoskeleton. Cytoskeleton is kind of an interesting addition to this list. In the old days, it was not usually considered an organelle, but in today's science, it is. So we'll start with nucleus, and some of you know that nucleus translated using the zoological roots means little nut. And we discussed the nucleus a little bit in the classroom, but we're going to expand on the nucleus and its responsibilities and functions here. First of all, there's an interesting thing about the membrane of the nucleus. The nucleus has a double membrane, sort of like a double wall in a house. So there's an inside membrane, an outside membrane, and a space in between. And the nuclear membrane tends to have very large pores. These are nuclear pores. Sometimes it's referred to as a nuclear envelope. And we'll see that it connects to some of the organelles in the cytoplasm. So the endoplasmic reticulum is actually continuous with the nuclear envelope or nuclear membrane, and this facilitates transfer of material between these two structures, between the nucleus and the endoplasmic reticulum. Here is a picture using a microscope of an amoeba, a single-celled organism with a nucleus. Amoeba are not particularly symmetrical in their shape, and in fact their shape changes as they move, but the nucleus is usually found pretty close to the middle. So what I've done, or at least I've tried to do, there may be some cases where this is not true, but mostly when I come to a function I'm going to put it in red. So the nucleus is especially important. It's sort of the control center for the cellular activity, and it's where most of the DNA is found. We're going to see that there is some DNA associated with the mitochondria, but that's different DNA than the normal nuclear DNA. The DNA is sort of like a cookbook with recipes in the DNA. It is where RNA is formed. And RNA and DNA were both talked about when we did the chemical lecture, which was topic 2. RNA is sort of a photocopy of sections of the DNA. Protein synthesis is going to be directed by RNA. If you'll recall, there were 3 kinds of RNA. Pants, do you know what they are, Pants? Right. Messenger RNA, ribosomal RNA, and transfer RNA. And there is material within the nucleus called chromatin. This chromatin coils and condenses near the time of cell reproduction. And when it condenses, it's referred to as a chromosome. So they coil to form the chromosomes. Chromosomes are, I'm pretty sure most of you have heard of chromosomes. Excuse me. There is an endomembrane system. And the endomembrane system includes a number of organelles that are actually connected and sort of interact with one another. And these are responsible for synthesizing and then releasing products. And so the nuclear envelope is part of this system. I mentioned that it is there's a space. It's a double membrane. The endoplasmic reticulum. Endoplasmic reticulum forms networks with the Golgi complex. The Golgi complex is sort of like a post office. It packages the products that the endoplasmic reticulum forms. The endoplasmic reticulum you could think of as a factory. The factory is where proteins, for example, are formed, and these proteins are formed with directions that came from the nucleus. We have lysosomes. Lysosomes are packages of mostly enzymes that are used to break down materials. And then vesicles. Vesicles are sort of, a vesicle is a generic structure, sort of like a little balloon that has material inside of it, and the material is determined by whatever the endoplasmic reticulum formed and the Golgi complex packaged for us. So we'll now talk a little bit about the endoplasmic reticulum. I abbreviate it with ER. It just makes it easier. It's kind of a long name, and ER is a whole lot shorter. But the endoplasmic reticulum is folded back and forth. The space inside of the membranes is going to be continuous with the nuclear membrane. I mentioned that already. They communicate. And it's also continuous with the space within the Golgi apparatus. The Golgi apparatus also is a series of membranes with a space in it. So this is sort of the manufacturing center of the cell. This is where materials are produced, both lipid and protein. There are 2 kinds of endoplasmic reticulum. There's the rough or granular endoplasmic reticulum, and rough is actually Pansy's favorite word. She's rolling her eyes at that. She doesn't have as sophisticated a sense of humor as most of you. The rough texture or appearance is caused by ribosomes, and the ribosomes are found on the outside of the endoplasmic reticulum, adjacent to or in contact with the cytoplasm. The ribosomes we're going to see are mostly RNA. There is some protein associated with them, and that is where protein synthesis occurs. And if you think of the endoplasmic reticulum as the manufacturing center for the cell, the ribosomes are the workers that actually produce the proteins or synthesize the proteins. So protein is going to be produced and go into the interior of the endoplasmic reticulum, and then the proteins are modified and processed. Some of them are incorporated into the membrane itself. Some of them are processed for export. You might wonder why would the ribosomes benefit by being outside of the endoplasmic reticulum? The answer is that the amino acids needed for the protein synthesis are in the cytoplasm. So that's the raw material that the workers need in order to get the finished product. The endoplasmic reticulum is connected to the nuclear membrane and on one side, and then it's connected on the other side to the smooth endoplasmic reticulum. We're going to talk about the smooth endoplasmic reticulum next. So, smooth endoplasmic reticulum is where phospholipids and lipids are synthesized. Also, it's where glycogen is synthesized and stored. Glycogen synthesis and storage primarily occurs in the liver and in the muscles, skeletal muscles. It's a storage of surplus energy that can be used when energy is needed and in short supply. And so it gets broken down once the glucose levels drop too much. Ribosomes are not employed in synthesis of these products, the phospholipids, lipids, or glycogen. So here's a photomicrograph of the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And you can see these little granules, if you will, or little particles attached to the endoplasmic reticulum. Those are the ribosomes. And you see with the smooth endoplasmic reticulum, there are no ribosomes. So here is the rough endoplasmic reticulum with the ribosomes on the outside. You can see that it is connected to the nuclear envelope. The lumen is continuous from the nuclear envelope to the lumen of the rough endoplasmic reticulum. And then the rough endoplasmic reticulum is also connected to the lumen of the smooth endoplasmic reticulum over here. Okay. So another interesting evolution of the endoplasmic reticulum for a specified or a specific function, a specialized function, is that in skeletal muscle cells, the smooth endoplasmic reticulum gets modified into something called a sarcoplasmic reticulum. Sarcoplasm refers to muscle cell cytoplasm, and the sarcoplasmic reticulum stores calcium until it's needed, and then during muscle contraction the calcium is released, and the calcium enables the cross fibers within the muscle to move and contract. And we'll talk more about that when we get to skeletal muscle. But that's where it's stored, and then once the contraction is over, the calcium is pumped back into the sarcoplasmic reticulum until it's needed again. Liver cells, the smooth endoplasmic reticulum appears to be involved in modifying some toxic materials, including drugs. These materials are altered and become less toxic, and then typically they're released back into the blood and excreted in the urine by the kidney. Excretion is when you get rid of a waste product, typically, as opposed to a secretion, which is something that you use. So it appears that the endoplasmic reticulum has a lumen that's continuous with the lumen of the Golgi complex. And I already mentioned the Golgi complex earlier as being sort of like the post office. This is where things are packaged for export. The export actually could be to the cell itself, or it could be released and go into the blood or the lymph for some other destination within the body. The endoplasmic reticulum forms vesicles in some cases, and sometimes the Golgi apparatus can form vesicles as well. Vesicles, I think you can think of as little balloons full of material that were produced in the endoplasmic reticulum. So those balloons could be holding enzymes, or they could be holding a neurotransmitter, or they could be holding some other material needed within the cell. The Golgi apparatus sort of has a shape like a lens, sort of curved a little bit, and there are a number of these sacs that vary in number, 3 to 20 on average, and they look a little bit like the endoplasmic reticulum. But it has three really important processes that it's responsible for, functions. It processes materials from the endoplasmic reticulum, it packages them, and then in some cases it secretes what the endoplasmic reticulum formed. So the enzymes in the Golgi apparatus can modify the lipids or proteins produced by the endoplasmic reticulum, and these may be transformed into glycoproteins or glycolipids. And as I said, these get packaged into vesicles, and the vesicles typically are released. So here we have a photomicrograph of the Golgi complex, including vesicles. Remember that it's kind of got a lens shape. Here's the lens shape. And then here are these little balloons that are coming from this lens, and those balloons are the vesicles, and they would have material that was produced by the endoplasmic reticulum. So the vesicles you can see being released, and those spheroids are the vesicles. Oh, by the way, vesicles is in red. It's not a function. It's an object. But I wanted to draw attention to it because it is listed sometimes as structures found within the cell. Okay, the bottom area in that lens-shaped structure would be where materials were being incorporated, and these come either from the endoplasmic reticulum, well, typically they do, and then they may be made into packets, or packaged into packets, I guess would be a better way to put it. The materials are going to be either proteins or lipids typically, and if the material is going to be secreted from the cell, the vesicles actually move to the cell membrane and they sort of burst like a bubble popping as it comes up through water. So here's an example. Here we have the cell membrane. Here this vesicle had been released a little while ago from Golgi complex, and here is a protein, let's say it could be an enzyme, and it's going to be secreted by the cell, maybe a cell in the stomach or the pancreas. And then there's the fluid surrounding the cell. That would be called interstitial fluid. Here the process is proceeding. The bubble is popped completely at this point, and the vesicle is about to be ejected. And from the interstitial fluid, it could diffuse into the blood or into the lymph, or it could be used in the area that the secretion is being formed and released. So here we have the protein is completely ejected from the cell. Okay. What we just looked at is called exocytosis. So this material is leaving the cell, and eventually the material that's secreted may get to where it's needed, and usually at that point it becomes activated. It's not uncommon to have something secreted that's not active immediately, that it requires some further action. So a good example would be pepsin. In the stomach we release a precursor for the enzyme pepsin. It's called pepsinogen, and when the pepsinogen is in the presence of low pH, a very acidic environment, it gets converted into pepsin, and then we use that pepsin to digest proteins. Other vesicles actually take whatever they contain to somewhere within the cell, and there they would burst and that material would be available, or they might be stored. A good example of vesicles that are stored for later use within the cell are lysosomes, and the Golgi complex produces these. They contain enzymes, digestive enzymes that are really strong, hydrolytic enzymes, and these are used to digest old organelles that are no longer functioning properly. We find a lot of lysosomes in white blood cells. White blood cells use these lysosomes to digest foreign organisms or foreign materials. So here is a photomicrograph of a lysosome. They actually attack the materials that the white blood cells have engulfed. We're going to see endocytosis not in this Camtasia, but in the regular lecture when we talk about the cell membranes. So not going to go into it. But endocytosis is essentially running a movie in reverse of exocytosis. The particle would be outside, the cell would sort of surround this particle, and then it would be incorporated into the cell. And these lysosomes could attack whatever it is that the white blood cell has engulfed. They're also important in tissue reduction. So sometimes you may have a wound or damaged cells, and these molecules or cells can actually be metabolized by the cell. A good example might be if you had a hemorrhage and you had a black and blue. The red blood cells that are in the tissues are basically damaged cells, and they can be digested, and gradually the black and blue should go away. Peroxisomes, they're sort of like lysosomes except they're smaller, and they have specific enzymes that have a couple of really important functions. One is they detoxify alcohol, they detoxify certain kinds of drugs, and also some organic waste. And these wastes are formed during normal metabolic activity, and the peroxisomes allow us to make them less harmful, and we can usually get rid of the material that is the result of that breakdown. Here is a peroxisome. So the liver and the kidneys are places where we have a lot of peroxisomes, and maintaining health of the body fluids and by getting rid of materials in the tissues. Another thing the peroxisomes can do is digest fatty acids, so that cells can metabolize the fragments that are available after that digestion occurs. We use fatty acids as an energy source when glucose is not available. Another thing that they do is they convert hydrogen peroxide, which is mildly toxic, into oxygen and water. And oxygen and water, neither of them are toxic, so this takes a toxic material and makes it untoxic or not toxic. One of the sources of hydrogen peroxide in the body is when white blood cells break down foreign material. Very sophisticated scientific word for foreign material is foreign stuff. Vacuoles. Vacuoles are sort of like lysosomes, except they are usually formed not at the Golgi complex but at the surface of the cell membrane. And vacuoles are used to isolate materials or objects inside the cell. They're frequently involved when you have pinocytosis, or drinking by the cell, when the cell engulfs something that's external to itself. We'll talk about that. Actually, one of the cases of vacuole formation would be when white blood cells engulf foreign organisms or materials. Another actually occurs in the stomachs of infants. When infants consume milk, some of the materials are engulfed by the cells lining the stomach. Mitochondria. Mitochondria are really an interesting organelle. Mitochondria is plural, mitochondrion is singular. These are sometimes called the powerhouse of the cell. This is its function. The mitochondria are organelles that are really good at forming adenosine triphosphate, or ATP. And during normal breakdown of carbohydrates, about 95% of the ATP produced from that breakdown is formed by the mitochondria. Here's the interesting thing about the mitochondria. There is very strong evidence to suggest that mitochondria were not part of the original animal cells, but rather an organism that invaded the cells. And the cells and the organisms have eventually worked out a mutually beneficial relationship. The mitochondria get fed by the cells, and the mitochondria are way better at making ATP, adenosine triphosphate, than the cells are. So it's it's a way of increasing our efficiency, and increasing efficiency is really important. In terms of the breakdown of lipids, typically 100% of the ATP generated by metabolizing lipids occurs inside the mitochondria. This is a really cool thing about mitochondria, and that is that the number of mitochondria per cell can be actually adjusted to best deal with the level of cellular activity. So an example, if you have somebody who's a real couch potato and they never exercise, the number of mitochondria in their muscle cells will be relatively low for that person. If that person then starts to go on to a predictable schedule of exercise and they are physically active, the number of mitochondria actually increase. Another interesting thing about mitochondria is that if you look at the world's elite endurance athletes, people who run marathons or compete in Ironman triathlons and things like that, they tend to actually have more mitochondria per cell than normal people, even if the normal people are trying to compete in the same events. And this really, if you think about it, is sort of what we sometimes refer to as natural aptitude. People who have a lot of mitochondria, more than the normal person would have, are going to be better at endurance activities because they can release energy more efficiently and increase their performance. So here's the reaction that occurs within the cell when the cell requires energy. The ATP molecules, adenosine triphosphate, combine with a water molecule, and in doing so they split. You end up with an adenosine diphosphate molecule, a phosphate group that broke off, and energy is released. This energy is what we use basically for three activities. We use it for mechanical activity of cells. We use it for transporting material through the cell membrane in some cases, not all cases, but some cases, and we'll talk about that in topic 3 in class. And then we also use that energy for cellular synthesis of organic materials. Typically energy is required when you make an enzyme or you make pretty much a protein or a lipid of any sort. So mitochondria have a double membrane, and when we look at the mitochondria, not only does it have a double membrane, but it has these shelves that extend out into the volume of the cell. And these shells are referred to as cristae, and they increase something. What do they increase, Pansy? Pansy knows the answer to this. Right. Surface area to volume ratio. So by having these shelves, there's way more surface area than there would be if it were just a big bubble. We're going to see this kind of shelf projection kind of a thing also in the small intestine, where it increases the surface area of the small intestine for absorption. In this case, it increases the surface area where the enzymes can be found that break down the molecules that are inside of the mitochondria. So the cristae project into the matrix. The matrix has a gel-like fluid surrounding these cristae. And the interesting thing, I remember I mentioned that mitochondria appear to be actually a foreign organism that invaded animal cells a long time ago in the development of animal life on our planet. There is mitochondrial DNA, and the mitochondrial DNA is different from the nuclear DNA. This is one of the pieces of information that suggests it was probably a foreign cell. So ATP synthesis, that is forming ATP from ADP and a phosphate group, requires that we have oxygen. You can actually produce a little bit of ATP without oxygen, but not very much, and you get some byproducts that are sort of not very good for the cell. So you really have to have oxygen. This is why we have to breathe in order to survive for long periods of time. And when that breakdown of organic materials using oxygen is accomplished, carbon dioxide and water is produced. And I think we talked about that back in the chemistry section. This process of breaking down organic molecules in order to release energy and form ATP is called cellular respiration. And a really key thing here, you want to really keep this in mind, we don't make energy. We release energy from the molecules that we consume and are able to get into our bodies. But we don't make the energy. The energy is already in those molecules in the form of bonds. So, as I mentioned, the enzymes are going to be on those shelves, and the shelves increase the surface area, which means there are more enzymes to break these things down that you are able to get into the mitochondria. Then, once the ATP forms, it diffuses from an area of high concentration to low concentration. It should be in high concentration within the mitochondria because that's where it's being produced. It's going to be in low concentration throughout the cell where it's being used. So it basically moves from where it's formed to where it's used. There's a net flow from high to low. Next, we have a nucleolus. Nucleolus is one of the non-membranous organelles. It's an area associated with the nucleus. It's inside the nucleus. And we already know that nucleus means little nut. Nucleolus means a wicked little nut. This is where ribosomal RNA is formed, and it's also where ribosomal RNA is stored. And ribosomal RNA is going to be essential. That's what it makes up the parts of the granules that we saw in the endoplasmic reticulum. So that's responsible for protein synthesis. So ribosomal RNA, real important for the ribosomes. Ribosomes, these are also non-membranous organelles. The ribosomes are the workers. Remember at the manufacturing plant, the endoplasmic reticulum, and the ribosomes construct proteins. So they don't do this randomly. They actually read, if you will, the directions that are contained on the messenger RNA molecules. And messenger RNA is sort of captured by a ribosome, and then the ribosome reads it almost like a zipper. It goes along the length of the messenger RNA, gets the code from the messenger RNA, and then selects amino acids in the appropriate sequence to form the proteins. So the ribosomes are going to be on the rough endoplasmic reticulum. In fact, that's why the rough endoplasmic reticulum looks rough. So here we see rough endoplasmic reticulum, and we see the ribosomes all along the outside of the endoplasmic reticulum's membrane. On both sides, remember it's a double membrane. Okay. So there are also some ribosomes that float around inside of the cytoplasm. Sometimes cytoplasm is called cytosol. I'm the messenger here. This isn't my idea to have multiple names for all of these things, but that has happened over time. Some physiologists refer to these things with different terms or slightly different terms. And the ribosomes that are in the cytoplasm are also going to produce proteins, and they may stay in the cytoplasm, or they may actually enter the organelle. That is the proteins, not the ribosomes. So there are two subunits for the ribosomes. One of the units is a protein, and one of the units is the ribosomal RNA. We have a centrosome. Centrosome is going to be found near the nucleus. It has centrioles, and we're going to talk about centrioles in the other portion of the lecture for this topic, the part that we'll cover in class. So I'm not going to go over centrioles now. We'll talk about them there. But the centrosome actually does a couple of pretty important things. One of the things that it does is it regulates the cell cycle progression. That is, the cell cycle is when it goes through mitosis. We have interphase, and then prophase, and metaphase, anaphase, telophase. So it regulates that progression, make sure that it proceeds in the appropriate way. And it also organizes the, it's a main organizer, if you will, or organizing center for microtubules within the cell. We also have cilia. Cilia are typically found in groups. They're not singular or solitary. So cilia would be the more normal term to use. Cilium is a singular term. And these are going to have centrioles in them. Remember, we'll talk about those in class. In single-celled organisms, ciliates, the cilia are responsible for propelling the organism through the water that it lives in. They're sort of like oars in a boat with multiple people rowing the boat. Some of you have probably seen movies where in the old days boats were sometimes rowed often by people who were not paid very well or were slaves. Even today you can see this sort of arrangement if you watch those races with the people in the Olympics rowing those racing shells. So in a single cell organism, it would propel the organism just like oars through causing the boat to move through the water. In a multicellular organism, though, the cells are fixed, so the cells can't move. And when the oars, or when the cilia move, they push the material adjacent to the cell. So here is a drawing of an individual cilium. We have the plasma membrane of the cell being continuous with the membrane that covers the cilium. And then inside there are 9 sets of microtubule doublets, and there's a central pair as well, and these are responsible for the movement of the cilium. The really cool thing is that they're coordinated so they all push in the same direction. And here we have a cilia, an individual organism with multiple cilia, and you can just make out these little hair-like projections coming off the cell. Those are the cilia. There are basically 3 ways that single cells move. One is with cilia, one is with flagella, which we'll talk about next, and one is with amoeboid movement. Of the 3 modes of transportation, the ciliates move them most quickly. Inside the cell you can see there's a nucleus right there, and you can see some little balloon-like structures. Those are vacuoles, most probably. Okay, so where do we find these cilia? In humans, we find them in the respiratory passages, the oviducts, and the ventricles of the brain. And the cilia are anchored by a structure called a basal body, and that's in the cytoplasm, and then it does have those microtubule doublets running through it. So in the respiratory passages, the cilia move mucus, and in the oviduct or uterine tubes, they move the ovum toward the uterus, and then in the brain they move the cerebrospinal fluid. Cilia are typically shorter than the flagella. As I mentioned, mucus moved in the respiratory passages. In the reproductive tubes, the uterine tubes, they move the ovum. And then, as I mentioned, they're that outgrowth of that basal body, which is pretty similar to a centriole. Again, centrioles will be discussed in a regular lecture. I should mention something about this, though. People who smoke lose the ability of having their flagella in the respiratory passages work. They stop working, and this is not. This is one of the unfortunate consequences of smoking cigarettes, and that is that the mucus then will move inferiorly because of gravity, and so the mucus will end up going down into the lungs, and you're really better off not having that happen. The good news is that if you give up smoking, the cilia will recover and move the mucus like they're supposed to move the mucus away from the respiratory passages and into the pharynx. Another unfortunate thing about this is that you don't have to be the person smoking the cigarette to have this happen. If you're in the presence of somebody smoking, you have the same problem occur. The cilia, as I mentioned, are coordinated. They have a uniform beat, so they all push in one direction. And they are connected by accessory proteins, dynein arms within the cell. There are some proteins holding the microtubule doublets together. There are some accessory proteins that split ATP, and when that happens, it causes the cilia to bend. And as I said, they all bend in one direction. The coordination of how that happens isn't really well understood, but it is obvious that it occurs. Should I tell you where you can see cilia? If you take a quahog or a hard shell clam and open it up, assuming it's still alive, if you look carefully at the gills and you watch it kind of in the light, you should be able to see the cilia beating on the gills. It's kind of a cool thing to witness. So next we'll talk about flagella. Flagella is plural, flagellum is singular. This one, as opposed to cilia, the flagellum is normally a singular solitary structure as opposed to being plural. But it also has centrioles, and it's basically just a really long solitary cilium, although there are some species where there are multiple flagella on their spermatozoa. The flagellum is used by the spermatozoa to be able to swim. It propels the spermatozoa through the female accessory tubules. The male tubules, the spermatozoa, are pretty much pumped, but when they get to the female tubules, they swim. So here are some flagellates, and you can see the individual flagella here. Here's one here, and here's one here. It's a fairly good picture of them, actually. If I were to ask you to list organelles on a quiz, and that certainly is a reasonable thing to expect, and you put down a list, I would consider flagella and cilia to be one example. They basically are the same thing, they're just slightly different in terms of length and how many of them there are. So we have the cytoskeleton. This is sort of a new addition for cell organelles. These are filamentous protein structures within the cell, and they have a number of functions. They help maintain the shape of the cell. Cells have pretty much predictable shapes. They anchor the organelles within the cell so that they are stable in position, and in some cases they help the organelles move. There are basically three kinds of tubules or filaments that are included as the cytoskeleton. We have the microtubules, intermediate filaments, and actin filaments, and we'll talk about each of these in turn. So we'll start with the microtubules. Microtubules are hollow cylinders. They have globular protein. There are 13 rows of globular protein called tubulin, and they are controlled by the centrosome. Remember, we talked about the centrosome being in the nucleus, or near the nucleus rather. And this is in the same general area that centrioles are found. The centrioles become apparent when cells undergo cell mitosis. The microtubules radiate outward from a centrosome. Maybe a good example would be if you looked at some sea urchins have things radiating out from their bodies, and these would sort of be like centrosomes. As I mentioned, they help maintain the shape, and they also appear to form tracks along which the organelles move. The spindle fibers they're going to be important to move the chromosomes during cell division, and those involve microtubules. And we have intermediate filaments. These are tough and can be resistant to being stressed, and they form junctions between cells. So from one cell to another, especially important in the epidermis, connecting the cells of the skin. And then we have the actin filaments. Actin filaments are really important in muscle contraction. All three kinds of muscles, skeletal, smooth, and cardiac. These are long, thin fibers. They tend to occur in bundles, and they are involved in the contraction of the cells and movement. Here's a pretty cool thing. They can be assembled or disassembled, so you can actually add extra contractile filaments in muscle cells as you build strength. And conversely, if you don't use the muscle cells, these bundles start to diminish or dwindle in numbers, and they are disassembled. So your muscle cells get smaller. So this gets us through the part of the quiz 2 dealing with the organelles. I hope that you learned some stuff. Pansy's gone into a coma here because I haven't asked her enough questions. But you will be responsible for this on the quiz, and it would be really helpful for you when you're trying to address the wish list of cell concepts or concepts for topic 3. Okay.